Hello, welcome to today's lecture. Today we'll continue to talk about the functions of the complement. After activation of the complement system, and we will talk about what the functions of the complement. The biological consequences of the complement activation include the membrane attack complex mediated cell lysis and split products mediated activities, including the inflammation, opsonization, and clearance of the immune complexes. So first, the membrane attack complex mediated cell lysis. The final step of the three pathways of activation is to produce the membrane attack complex on the surface of the target cells. The complex inserts into the membrane of the target cells, and then the water and ions within the cell will cross the cell membrane, leading to the lysis of the target cells. And the target cells for MAC are mainly gram-negative bacteria, parasites, viruses, erythrocytes, and nucleated cells. So, complement is very important to fight against the infection. And as we know, during the process of the complement activation, a lot of the fragment will be generated. Some fragment will release into the body fluid. For example, C3A and C5A, they are called anaphylatoxins. They can cause the inflammation response. We have talked about mast cells and basophils in the blood. And these two cells contain granules. And within the granule, there are a lot of the inflammatory mediators. Once activated, those cells will release the inflammatory mediators to cause the inflammation. This process is called degranulation. And these two kinds of cells express a receptor, which is called complement receptor. Their complement receptor combined with C3A and C5A. So after their binding, the mast cells and blood basophils will be activated and induce the degranulation. So they cause the inflammation. C3A, C5A also can cause the allergic symptoms. In addition, C3A and C5A have the chemotoxic effect on the monocyte and neutrophils. That means they can recruit a lot of the monocyte and neutrophils to the inflammation location and increase the inflammatory response. And next activities mediated by the split products are opsonization. In previous, we have talked about opsonization mediated by the antibody. So today we will talk about the complement-mediated opsonization. C3b is the major opsonin of the complement system. C3b combined with the target cell or antigen-antibody complex. And it also combined with the receptor expressed on the cells, for example, the phagocytes, macrophage. And these phagocytes express the receptor bind C3b bonded target cells. And then those phagocytes can internalize the bacteria or other target cells and then degrade them. This is called complement-mediated opsonization. In addition to C3b, C4b and inactivated C3b also can have the opsonizing activity. The last activity mediated by the split products are clearance of the immune complexes. As we see that once the immune response takes place, we can generate a lot of the antibody, and those antibodies will bind with antigen, form the immune complex. The soluble immune complex must be removed from our body because the deposition of the soluble immune complex will cause the disease. So our complement are responsible for their clearance. This is the antigen-antibody complex. It is a soluble immune complex. And C3b combined with this immune complex. The other end of the C3b combined with complement receptor on the red blood cells. So red blood cells combined C3b binded immune complex. So red blood cells carry those complex. They traveled in the blood circulation. And then they will be carried to the liver and spleen, where the phagocyte located. The phagocyte can express more complement receptors and Fc receptors. So they can remove the complex from the red blood cell surface without destroying the red blood cells. Then they will internalize the complex and degrade them. That is the clearance of the immune complexes. That's all for today. Thank you for your attention.